Hey everyone, and welcome to today's Explainer. We are about to dive into what I honestly think is one of the coolest biological detective stories of all time. Today, we're going to explore the ultimate blueprint of life, the hidden code locked inside your very own cells. Yep, we're talking about heredity. Get ready, because we are going to completely unlock the master code of how traits are actually passed down from one generation to the very next. So, right off the bat, here's a mystery for you. Think about your siblings, or take a good look at your parents. You definitely share some similarities, right? Maybe you've got your mom's smile, or your dad's nose. But why don't you look exactly like a clone of them? Why is there literally always some sort of subtle difference? Well, that fundamental question is exactly what we're going to answer today.
Here is our game plan for this explainer. We'll start with the blueprint of variation, then check out inherited traits and earlobes. After that, we'll hit Mendel's rules of inheritance, the dihybrid cross, how genes express traits, and wrap it all up with how sex is determined. Okay, Section 1, the blueprint of variation. Let's think about how differences actually build up over time. You see, when a single organism reproduces asexually, like say a bacterium just dividing in two, the DNA copying process isn't absolutely 100% perfect. Tiny, subtle inaccuracies happen. And those little variations accumulate generation after generation. Now, throw sexual reproduction into the mix, and this diversity is magnified even further. It's kind of like a massive game of genetic telephone, where the core message gets slightly tweaked with every single generation, which ensures no two individuals are ever perfectly identical. Moving on to Section 2, inherited traits and earlobes.
I want you to literally pause for a second and feel your own earlobe. Seriously, reach up and touch the bottom of your ear right now. Does it hang down free, or is it attached directly to the side of your head? If you were to walk around and check all your friends, you'd see a total mix of free and attached earlobes. These are inherited traits. They're the real-world physical manifestations of that hidden code passed down from your parents. And by tracking simple traits just like this one, we can start to uncover the underlying rules of heredity.
Which brings us to Section 3, Mendel's rules of inheritance. Enter Gregor Johann Mendel. Now, Mendel wasn't just some monk quietly growing plants in a monastery garden. The guy was a brilliant scientist. Long before him, people totally knew that traits were inherited. But Mendel, he was the first to actually blend science and mathematics to prove how it worked. He rigorously counted thousands of pea plants, keeping exact track of specific traits generation after generation. So, let's look at exactly what he found out.
Imagine a blank whiteboard right in front of you. Let's sketch out Mendel's most famous experiment together. Step 1, he took pure tall pea plants and crossed them with pure short pea plants. Simple enough, right? But then, Step 2, in the very first generation, which we call the F1 progeny, every single offspring plant was tall. There were no medium height plants, just tall. It was like the short trait had just completely vanished into thin air. But wait, here comes Step 3. He let those F1 tall plants self-pollinate. And boom, in the next generation, the F2 progeny, the short plants suddenly reappeared out of nowhere. Exactly 1/4 of them were short. How in the world does a physical trait just disappear and then magically bounce back?
Okay, let's demystify this and break down that famous 1:2:1 ratio. Mendel figured out that each plant actually has 2 copies of a factor for each trait, one inherited from each parent. Today, we call those factors genes. Let's use some letters to make it clear. We'll use a capital T for tall and a lowercase t for short. If a plant gets a big T and another big T, it's tall. If it gets a little t and a little t, it's short. But what happens if it gets one of each, a big T and a little t? Well, when you run the combinations, a TT plant is tall. So when those F1 TT plants cross with each other, the math perfectly sorts out to a strict ratio of 1 TT to 2 TT to 1 TT.
The absolute key to this whole puzzle is understanding why that mixed T plant is tall. It's all about dominance. A dominant trait, like tallness, represented by that capital T, is super powerful. It literally only needs one single copy to express itself and totally overpower the other trait. On the flip side, a recessive trait, like shortness, with our lowercase t, is kind of shy. It requires 2 identical copies, a TT, to finally show up in the physical world. And that right there is exactly why the short trait could hide away in the F1 generation only to reappear in the F2.
Let's step it up in Section 4, the dihybrid cross.
So I challenge you to imagine this.
What if we track two completely different physical traits at exactly the same time? Mendel took a plant that was tall and had round seeds and crossed it with a plant that was short and had wrinkled seeds. What do you think happens here? Do tallness and roundness always stick together like some sort of genetic package deal?
Well, here is the mind-blowing realization. In the F1 generation, they were all tall and round. Our dominant traits were just flexing their muscles again. But in the F2 generation, a massive recombination occurred. Mendel suddenly found plants that were tall but had wrinkled seeds, and he found plants that were short but had round seeds. The big takeaway? Traits are inherited completely independently of one another. The trait for the plant's height absolutely does not stick to the trait for its seed shape. They sort themselves out entirely on their own.
Next up, Section 5, How Genes Express Traits.
Let's use a factory analogy to really visualize how this chain reaction happens inside you. Your cellular DNA is the ultimate master blueprint. A specific section of that DNA, which we call a gene, acts like an instruction manual for building one single protein. Think of this protein as an enzyme, basically a highly efficient factory worker. Now, if the gene is perfectly coded, that enzyme worker builds a whole lot of plant growth hormone, that's the product, and lots of hormone equals a tall plant. But if that gene is altered even slightly, the enzyme worker becomes inefficient. Less hormone gets produced, and the physical result is a short plant. So genes literally control your physical traits by giving orders to these microscopic cellular factories.
But hang on, how do we make sure the factory gets the right manuals from both parents without doubling the total amount of DNA every single generation? Well, DNA isn't just one endlessly long thread. It's bundled up into separate independent pieces called chromosomes. You've got two copies of each chromosome, one from mom, one from dad. But here is the real magic trick. Your germ cells, the reproductive cells, only take one chromosome from each pair. So when two germ cells combine during reproduction, the normal, healthy number of chromosomes is perfectly restored in the new child. It's a brilliantly balanced system.
Finally, Section 6, How Sex is Determined. Now, what's genuinely wild is that not all animals handle this the same way. In some reptiles, the actual temperature the eggs are incubated at completely determines whether the babies hatch as male or female. Some snails can even change their sex based entirely on their environment. But humans, we rely strictly on genetics. Specifically, we rely on the inherited X and Y chromosomes.
And here is the final twist of our biological detective story. Every single human child inherits an X chromosome from their mother, regardless of whether they end up being a boy or a girl. The mother literally only has X chromosomes to give. Therefore, it is exclusively the father's genetic contribution that ultimately determines the sex of the child. If the father passes on his X chromosome, the child is a girl, XX. If the father passes his Y chromosome, the child is a boy, XY.
Wow, okay, we have covered some massive ground today. Let's do a really quick rapid-fire summary to lock all of this in. Number 1, tiny variations accumulate over time during reproduction, and that creates diversity. Number 2, thanks to our guy Mendel, we know traits are passed down and they can be either dominant or recessive. Number 3, the dihybrid cross proves that characteristics are inherited completely independently of each other. And number 4, in humans, sex is genetically determined specifically by the paternal X or Y chromosome. You have officially mastered the foundational code of heredity.
Before we go, I want to leave you with a pretty profound question to chew on. We've just seen how DNA copying isn't perfectly identical, and how tiny variations accumulate with every single generation. If you take that process and multiply it over thousands or even millions of years, how might those incredibly small inherited traits build up to drive the evolution of entirely new species? Think about that. Keep exploring the microscopic world, and I will see you in the next explainer.
All right, welcome to this explainer. Today, I am literally putting on my veteran KSEAB board evaluator hat to show you exactly how to secure every single mark in Chapter 8, Heredity. If you're gearing up for the highly competitive Karnataka SSLC board exams, specifically the 83E science paper, you need to listen very closely. We aren't here to just recap what a gene is, right? You already know that. Instead, we are hacking the official marking schemes to figure out exactly how to put your knowledge on paper to get a perfect score. So let's jump right in.
Here is our strategic blueprint. We're looking at the blueprint to heredity marks, traits, and one-mark traps, cracking Mendel's crosses, the checkerboard cheat code, and scoring sex determination.
Section 1, Blueprint to Heredity Marks. Let's see what the examiner's eye is actually searching for.
Okay, so look at how your exam paper is actually broken down. As examiners, we categorize your answers strictly by value points. You've got to shift your mindset from just casually reading textbook paragraphs to aggressively hunting for exact points. We're searching for 1-mark multiple-choice definitions, 2-mark balance differences, 3-mark analytical crosses, and those big 4-mark structural flowcharts. Literally every half-mark is accounted for in the official guidelines, and you need to know exactly where they're hidden.
Moving on to Section 2, Traits and 1-mark traps. Let's tackle the high-frequency spots where students routinely drop the ball.
Take a look at this question. Why can't the experiences of an individual during its lifetime be passed to its progeny? Well, this is an absolute favorite for the examiners, pulled straight from the 2021 board paper. It looks like a conceptual question that needs a massive, winding paragraph, but honestly, that's exactly the trap. The official key answer requires incredible brevity. The only phrase the evaluator is scanning your page for is, because they are acquired traits. Seriously, if you write a huge paragraph about lifetime experiences, but fail to include those 2 magic words, acquired traits, you lose the mark entirely. Don't overcomplicate it. Just give them the value point.
Now let's look at a classic 2-mark direct question, differentiating dominant and recessive traits. According to the official August 2024 key answers, your definitions most perfectly contrast. A dominant trait is the one out of the 2 copies that expresses visible character more in progeny. The recessive trait is the one that expresses less or not at all. A super common student mistake is defining only the dominant trait and moving on. Keep your definitions balanced, almost like a chemical equation to secure both marks.
Section 3. Cracking Mendel's crosses. Time to escalate to the heavy-hitting analytical questions.
If you're asked to define a monohybrid cross, there's a specific definition that acts as your absolute gold standard. A cross between 2 plants which differ in one specific character. The official model answers explicitly award 1 full mark just for this concise sentence alone. Memorize it word for word. Don't add fluff.
Okay, now let's look at a very common textbook trap. A cross of tall pea plants with violet flowers and short pea plants with white flowers yields all violet flowers, but half are short. So what is the tall parent's genetic makeup? You know, students rush this and just assume the tall parent is completely dominant. But wait, read the prompt. Half the progeny are short.
That brilliant little deduction gives us the solution. The correct genetic makeup is a capital T, a lowercase t, and 2 capital W's. Why? Well, we use a capital T and a lowercase t because the parent absolutely must carry the recessive short gene if half the offspring are short. But we use a double capital W because the violet dominant trait expressed completely in all the progeny. This is high-yield analytical scoring at its finest. I need you to take a mental screenshot of this right now. Confusing phenotypic with genotypic ratios is literally the #1 student mistake in this entire chapter. So here's your high-yield memory aid. P is for phenotypic, and P is for physical appearance, what you actually see, which gives you the 3-to-1 ratio. G is for genotypic, and G is for the underlying genes, giving you the detailed 1-to-2-to-1 ratio. The board loves to test these in multiple choice questions, so lock these numbers into your memory for sure.
Section 4, the checkerboard cheat code. Let me let you in on a massive evaluator's secret.
This quote is pulled directly from the official KSEAB key answer marking scheme. 1 mark can be awarded if checkerboard is written for F2 generation. So what does this actually mean for you? It means when in doubt, draw the board out. Even if you completely blank out on the written explanation, throwing down a checkerboard acts as an immediate safety net for partial marks.
The standard grid layout for a red and white pea plant cross is exactly what the evaluator expects to see on your paper. Notice the clear labeling of gametes on the top and the side and perfectly filled combinations inside the boxes. But drawing the grid is really only part of the battle.
Here is where the marking scheme absolutely traps you. You get marks for drawing the box, yes. But if you forget to declare those ratios, the phenotypic 3-to-1 and genotypic 1-to-2-to-1, at the bottom of your board, we legally have to deduct half a mark for each missing ratio. Never, ever leave a checkerboard without its ratios written out clearly at the bottom.
Finally, Section 5, Scoring Sex Determination. Let's talk about a guaranteed 3 to 4 mark structural breakdown. The standard diagrammatic flowchart for human sex determination is visual proof of inheritance and an absolute goldmine for scoring structural marks. It clearly maps out the crossing of the father's XY chromosomes with the mother's XX chromosomes to determine the gender of the zygote. It requires very little writing but awards massive points.
Let's look at exactly how we evaluators distribute the marks for that diagram. You get half a mark just for labeling the father as XY. Another half for the mother as XX. But here is the trap. If you draw all those beautiful crossing lines but forget to explicitly write the words girl and boy at the bottom next to the zygote combinations, you are throwing away a full mark. Label absolutely everything.
Let's see how this all builds into a complex, multi-part exam question. The board might ask you to contrast how plant height is determined versus how human sex is determined. This is a brilliant comparative setup from the model papers because it tests two totally distinct biological concepts at the exact same time.
To secure full marks here, you absolutely must show total mastery of both systems. For plant height, the required value points are mentioning that genes secrete a specific enzyme, and if it works efficiently, the plant grows taller. For the human male child, the absolute required value point is stating that the child receives a Y haploid chromosome from the father and an X from the mother. Structure your answer exactly like this, contrasting the two clearly, and the evaluator will gladly hand over the marks.
As you head into your exam, stop thinking like a student and start thinking like an evaluator. Remember the golden rules for heredity? Write the ratios, label the gametes, and when in doubt, draw the boards. If you execute these strategies, those 80 out of 80 marks are practically yours for the taking. The marking scheme is no longer a secret. So are you ready to stop just casually reading the textbook and start actually hunting for those value points? Good luck out there, and I'll catch you in the next explainer.